I'm speaking on behalf of a group that's called Animal Physiology and Ecology, but really, as you'll see, it's a much more eclectic group of projects than that. And I'm going to speak for a few minutes and hand it over to Adam Britton. Some of the key staff in this group is Keith Christian. I work mostly on amphibians, physiology, amphibians and reptiles, the ecology and physiology, and also microbial ecology of animal skins and translucent rocks, and I'll explain that a little bit more. Christine Schlesinger is in Alice Springs. Her work is around the effects of buffalo grass on vertebrate ecology. Dr. Wenkang Ping, who's interested in biological control on tree crops, and after 22 years on soft money, Dr. Ping is retiring this year. Carla Eisenberg is working on turtle ecology and conservation, in particular on an endangered species of turtle in East Timor. And Chris Tracy, who used to be here, I'll mention him because he's still collaborating with me on amphibian and crocodile projects, but he's now at California State University in Fullerton. And then Adam Britton, he'll speak in a few minutes. Some postgraduate students, Matt Bryan is working on survival of saltwater crocodiles during their first year, which is obviously related to the crocodile farming industry. Odette Gutierrez is working on organic cashew industry in East Timor. Sam Godwin's working on the physiological ecology of mangrove snakes. Georgia Kosmala is looking at cane toad physiology, both right across their range in Australia and as well as in Brazil. Georgia is actually a University of Sydney student, but she's working in my lab. Chava Weitzman is looking at the microbes associated with amphibian and reptile skins, and Chava is a National Science Foundation fellow from the University of Nevada at Reno, and should be here later this year. Bruce Pascoe is working on arid bird ecology, and Adam McPhee is working on cultural and natural significance of the Strelo collection in Central Australia. Just a few case studies. This is Dr. Ping's work on cashews in East Timor, and he is sort of the wrap-up of that project. He produced a photographic guidebook to be used by extension officers and farmers to help them right throughout the entire cashew production cycle. It has a booklet of over 400 pages, 120 pages, 400 photos in that. So it's very descriptive, and we're in the process now of trying to get that translated into Tetum. Matt Bryan was actually graduating in May. Matt was really a model student. He published 6 papers before he submitted his PhD. He, as I mentioned, he was looking at the problem of death of a lot of the juvenile crocodiles during their first year. And as you can see, he's been very successful at maximizing growth during the first year. But actually, this picture shows him. He's leaning on the world's strongest metabolic chamber, and we were measuring metabolic rates of crocodiles up to 400 kilograms in this metabolic chamber. Another project that's really sort of some of the blue sky work was looking at hypolithic cyanobacteria. That is the cyanobacteria that grow underneath translucent rocks, and some of these kinds of rocks are shown, the clean versions there up in the top corner. It includes quartz and agates and prehnite and other translucent rocks that have a community living underneath them that survive and photosynthesize using the light that passes through the rock. So it's a very specialized sort of community. And the other group that's very interested in this kind of work is NASA, because these sorts of animals, these sorts of organisms are probably, if there was ever life on Mars, they were of this sort. We started this work a few years ago, and initially we identified 60 new species of cyanobacteria under 9 different rocks that we sampled. And so we've expanded that more recently to look at much greater number of rocks, and also the spatial distribution of these cyanobacteria over a 10-kilometer transect. And a future project that we'll be doing with Chava Weissman will be looking at cutaneous microbial associations on amphibians and reptiles. Particularly amphibians have a very interesting microbial community on their skin. We know a lot about them from work over the last 20 years on amphibians and reptiles in this area. We know a lot about their physiology and what physiologically, there are gills, so to speak, of the arboreal species are quite physiologically distinct from the terrestrial species, distinct from the aquatic species. And now we're going to expand that work and see how that relates to the microbial communities on their skin. Now talk a bit about Christine's work in Alice Springs. Her research focus has been looking at how buffalo grass may have affected or is affecting vertebrate animals, or animals, not just vertebrates. She started with vertebrates, but animals in Central Australia. And several years ago, Christine established a series of buffalo-free zones, and you can see this square or this rectangular plot there where the buffalo grass was removed. And she's been studying those and comparing it to the buffalo grass areas, looking at, initially started with amphibians and reptiles, then she expanded to ants and birds, and most recently she's looking at the behavior of animals within those different zones. Some of the Alice Springs research students, I've already mentioned the first two on this list, the postgraduate students, but Christine also has a couple of honor students, one working on the Great Desert Skink, which is shown there, and also another student working on river red gum communities along ephemeral river corridors. Future work in Alice Springs, one project that's just getting started is looking at the threats to bilbies in the Tanami Desert, and this work is being funded by the Twin Bonanza Gold Mine. And then another project looking at invasive grasses and how they disrupt the plant-animal interactions and threaten the persistence of the semi-arid woodlands in Central Australia. So I'll pass over to Adam.
Thanks, Keith. I'll try and keep this brief. I'm one of the 90-odd adjuncts that Andrew was talking about earlier, and I'm working on a few collaborative projects with RIEL. And I wanted to talk about this particular one. It's something that we're also collaborating with Imperial College London, with a guy called Dr. Simon Pooley, because we're interested in how you mitigate risk when it comes to crocodiles, where of course that question is particularly relevant. Because if it comes to when you're trying to conserve crocodile populations in the wild, you've got to try and do everything you possibly can to stop those crocodiles from eating the people who live around it, otherwise you've got no chance. And so how do we actually do that? Well, what we've been trying to do is collect as much information as possible about crocodile attacks, and it turns out that there's actually a heck of a lot of that information out there, but it's spread out all over the place. So we put together in 2013 this thing called CrocBite, which was an online database where we could basically summarize all the information, create a database, an online database. People could go and add information which we would vet. And we've now got about 3,000 records in that. So we've got a huge amount of data, and it goes all the way back to the mid-19th century. But most of the data actually has come from the last few years. But when you've got all that amount of data, the next question is, how do you actually make sense of it? So as you can see, there's a lot of crocodile attacks around the world. Now, we have, at the moment, a very robust search facility, so you can go and you can filter the data to get to the country and the species and whatever that you're interested in. And once you've got that, then the next stage is we're going to start developing these analytical tools so you can actually do some fairly basic and hopefully more advanced statistical analysis on the data that you've actually filtered. And the other aspect of it as well is not just the analysis, but also the communication of this information to a variety of different audiences, whether you're talking about school kids or the media or people in general or government departments or whatever. And so, let me just try if I can get this working. This is one of the things we've been working with Dr. Pooley This is basically an organisation in the UK that specialises in creating infographics. So we gave them this challenge: how do you create infographics? So we used some data from South Africa and Swaziland. And it was essentially just a question of saying, okay, let's try and present information that's useful for people in easy to digest snippets. So here we have, how do you visualize where attacks are taking place and the intensity of those attacks, risky areas where you're more likely to find crocodiles, how the attacks what the relationship is between environmental variables, what the relationship is between the victims. You can see here most of these victims are local, and most of those locals are being attacked while swimming and bathing. And, it goes on and on. There's a lot of this stuff which I'd love to spend more time talking about. But let me see if I can get back to this. This is the actual site itself, and if you go and type crocbite one word into Google, you can actually go and find it. You can go and have a look. We don't have all of the stuff implemented yet, but it's coming very soon. And because of the modular nature of the site and what we've been trying to do, we've been getting small amounts of funding, and also we're going to be using crowdfunding in the future as well, just to try and add these modules to the site. And we're going to be working as well more with RIEL to try and get more work going on in the region, and hopefully get some more students involved in the process as well.